Welcome everyone. Visiting us annually. We love Andrew. I have a very long biography of him, which I was warned that I should cut short because he needs all the 9 lecturing time that he can have. First of all, again, we love Andrew. Thank you for coming again. Our natural sciences course, our core curriculum becomes much better than it already is with your contributions. So thank you very much. Andrew received an undergraduate degree in zoology from Oxford University. Then he moved to the US for graduate work at Princeton University in Marty Friedman Lab. His graduate study was about the way of detecting positive selection, adaptation, and Drosophila DNA sequences. He continued studying the same research topic with Dick Leventon at Harvard University. He is still at Harvard University as a lecturer in organismic and evolutionary biology department. His interests, obviously, number one, Sabancı University. Number two, genetic and statistical approaches to detecting adaptive evolution in genomes. Number three, he has a particular interest in history of science, especially interested in life and research of Alfred Russel Wallace. He delivered a wonderful lecture a year ago, 2 years ago, about him, who is the co-discoverer of Darwin of the theory of evolution by natural selection. He currently teaches what is life, evolutionary biology, understanding Darwinism, genetics, genomics, and evolution. He's also teaching NS 101 Science of Nature at Sabancı University. We're very proud to have him here. He has many publications, to name a few significant ones. Infinite Tropics from Vertebrates, 2002. DNA with Jim Watson, 2003. Penguin Classics edition of Wallace's Malay Archipelago. He has a lot of public talks, and he's a great public lecturer. I'm not going to list the talk. I'm just going to show you one example of such a talk right now, if that's okay with you.
Thank you very much, Emre. And thank you all for coming. I'm really pleased to see that Koç High School is here and Tivitol is here, 2 deep rivals. So I hope you don't end up fighting each other or something unpleasant like that. But anyway, it's great to see you. And thank you, Emre. It's the best part of my year. I mean, I live in America, for crying out loud. So it's my best part of the year when I get to leave the United States and come to Istanbul. One, because, yeah, this is the best city in the world, bar none. There is a problem that I'm at Sabancı University, which is nearer Ankara, I think, than Istanbul. It certainly feels like that when I'm stuck in the traffic. But the best thing about coming out is, and Emre's right, it's embarrassing to admit it, it's been 25 years. I taught the first class of students at Sabancı University who were, and this is the challenge for the current generation, the best students I've ever taught anywhere. And I've taught at Oxford, elite institution, Princeton, elite, snotty institution, Harvard, very elite, extremely snotty institution. There's some pretty smart students at those places. Anyway, I love this institution and it's a privilege to be here. But I'm not going to spend too much time talking about that and about the delights of Turkish food and God knows what else that brings me over here. I'm going to talk about what I think is one of the most interesting problems in evolutionary biology. No, one of the most interesting problems there are. End of story, right? This is not unique. This is not a peculiarity, a weird obsession of evolutionary biology. This is about us, humans. Remember, life originated maybe 3.9 billion years ago, and it was just a simple little chain of molecules, right? A single molecule, actually, right? RNA, wiggle, 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 self-reproduce, self-reproduce, 3.9 billion years ago. And then a process took over, the process we call evolution by natural selection. Natural selection kicked in straight away. There were competing wiggling molecules, both trying to assemble themselves most effectively. They're competing. The one that is better at harnessing the resources, acquiring the stuff, so to speak, for replication, is going to be much more successful. That's the one that's going to pass its traits on to the next generation. So it's super simple at this early stage, but natural selection is happening. And it continues to happen, and it's happened for 3.9 billion years. And it's an extraordinary, sobering realization that we are just one of the outcomes of 3.9 billion years from that RNA molecule, wiggle, 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 great apes, then us. We're just modified great apes. That is the huge message of this process, evolutionary biology, right? We were originally RNA molecules a few billion years ago, and now over time, we have transformed, if you like, through the evolutionary process into us, wonderful, brilliant, brainy us. But we are just modifications of what came before. Now, how has that happened? Well, the key to this process is genetics, right? Because it's genetic material, it's DNA that is being passed on from each generation. It's on genes, on DNA, on genetic material that natural selection acts. So there've got to be genetic differences, one that makes the zebra run fast, and the other which causes the zebra to run very slowly, right? Genetic differences, this one is going to survive, pass on the run fast genes, and the poor, sad, slow, fat zebra is going to be eaten by the lions and will not pass its genes on, right? It's genes that we're talking about. Now, we talk about differences in genes, and everyone in this room basically is the same set of genes, and there are a few tiny differences. You have an A where this person has a T at position 97 on gene 3 on chromosome 12, right? Tiny differences. And no, they're important. They account for all the differences we see in this room, differences in hair color, eye color, all sorts of other things, right? But that's kind of the icing on the cake, isn't it? Just changing a G or a T or a C, a single base pair here or there or whatever. Where do new genes come from? You're human because you have very different genes from most fish. You have very different genes from most insects. So how does the evolutionary process, it doesn't just change the odd letter A or G or C, how does it actually create whole new genes, whole new pieces of genetic information? That's what I want to talk about today. I'm calling it genetic novelty. And there are three basic mechanisms whereby the genome is dynamic, one of which I've already talked about, simple mutation. So this is, I love this plot. This is from a paper not that long ago. This is measuring the number of mutations in you as a new human being, okay? What we've done here is we've taken trios. This is mum, dad, and junior. We've sequenced the DNA, the total DNA, the genome of mum, the total DNA, the genome of dad, and junior. And there are going to be some mutations in junior which are not present in dad and not present in mum. Those are new mutations, brand new mutations, which have occurred in the generation of the sperm and the egg that made junior, right? And it's about 70. So every single one of you has 70 new, well, born with 70 new mutations. So that's actually quite a large number, if you think about it. It's also not a very large number if you think about the size of the genome. The genome is 3.5 billion base pairs. That's a lot of A, D, G, C, 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 G, G, C, 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 T, 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 A, G, right? I could do that for a very long time to get to 3.5 billion. So 70 is small, relatively speaking, but in absolute terms, that's a pretty high error rate for every new individual that's born. The other thing that's really cool about this is you can pass those mutations into whether they came from mum or dad, and dad is the issue, okay? Now, interesting, and also it's also most flat. Dad's age matters. Notice there's quite a slope here. Now, why are dad and mum so different? It's because of the different ways in which we generate gametes, eggs and sperm, basically. A woman, as a baby, is born with basically all the eggs she's ever going to have, right? So there's not much cell division required. Males are constantly creating new sperm, lots of cell division, lots of cell division, lots of cell division. Now, why is that critical? Well, where do these mutations come from? They're just copying errors. So if you've got 3.5 billion base pairs in a cell that you need to copy, the molecular system has to go along A, T, G, C, 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 G, T, A, making that perfect copy. And mistakes happen. It's a thermodynamic inevitability, right? Those are mutations, right? So the more cell divisions you have, the more mutations you're going to have, okay? So anyway, women fortunately give up, thank you, menopause. Men, yeah, I'm thinking of, I don't know, some very old men, usually horribly wealthy and wrinkled, who keep reproducing. Anyway, message for everyone here, avoid reproduction with very old men, just, you know, as a rule of thumb in life. So that's one of three sources of what I'm calling genomic dynamism. The other is on a much larger scale. Sometimes there are errors, again, in creating sperm and eggs, in terms of the chromosome complement and the structure of chromosomes. This is just one possible thing that can go wrong. So here we've got two separate chromosomes, and for reasons, and say this is a mistake, but it happens. You've got these switched out bits. So you've got the outcome is two hybrid chromosomes, both blue and orange, instead of a straight blue and a straight orange. So that's shuffling genetic information from one genetic neighborhood, a chromosome, to another one. And look, famous example, this is human chromosome 2. It's a long, big chromosome. Chromosomes are numbered according to their size. So this is the second largest, it's number 2, the second largest chromosome in the human genome. Turns out to be the product of two great apes. These are chimpanzees, these are gorillas, and these are orang chromosomes glued together. Okay? So it's basically a process a bit like that, which has occurred in our immediate ancestry, reducing the number of chromosomes that we have relative to great apes. So that's our second mechanism, these big rearrangements. The third thing, are you paying extra, by the way? I'm just curious because you're so close. The third thing is a really weird one. All animal species, have essentially parasites in our DNA. These are small stretches of DNA that can make copies of themselves and park themselves elsewhere into your genome. Often these started off life as viruses. That's what viruses do. They invade the cell, park themselves into your genome and produce extra copies of themselves. So it turns out your genome, almost half of your genome, consists of dead copies of these parasitic agents. We call them transposable elements. Transposable elements, things that can transpose from point A to point B by copying themselves. That's kind of remarkable. We think of a genome as it's a set of critical information, but you've got these nasty little things that jump in and out. And just imagine if it jumps into the middle of something that's important, it's going to mess up the thing that's important, the thing that is functional. And just to give you an idea of how significant these things are, the most common form of these things in humans is something called ALU. Oops, sorry. This is the ALU sequence, give or take. It's about 300 base pairs long. And you have approximately 1 million copies of this in your genome. All right? And look, this is actually an amazing plot. This is showing, these are the human chromosomes. Here's our friend chromosome 2, which we've stained for ALU. So chromosome 19 is basically ALU, whatever. Not so much on chromosome 18. But look, this thing is mighty common. It's 10% of your genome. And there have been times when this thing has been very active, producing many copies which have parked themselves into genomes, disrupting the function of functioning genes and so on. So here we're looking at these are different kinds of transposable elements. Just let's look at the dark brown, this ALU one. This is the level of genetic divergence we see amongst the different copies. Remember, you've got 1 million copies of these things in your genome. So this is the level of genetic difference we see amongst them. And you've got a big peak here, so about 7% difference. So in other words, maybe 10 million years ago in our ancestry, these things went crazy and produced lots of extra copies. So you've got, it's like being in a rainstorm suddenly in the genome. These things, bing, 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 all over the place. These are our three mechanisms that mess up genomes, that create dynamic genomes. So we've got our tools. Let's talk about how we can create new genes. And some of these aren't that exciting, but some of them are really amazing. This one is, well, it's kind of interesting. It turns out nobody believed this could happen, but we now understand that some genes come from nothing. Okay? Now, what does that mean? Well, the genome is huge, remember? So you've got tons and tons of genetic sequence. Sometimes a mutation will occur in a useless, dead, defunct genetic sequence that turns it from useless, dead, defunct into something useful, functional, and worthwhile, right? So you've literally gone from no gene to some gene. And it's kind of interesting. This is a famous molecular biologist, one of the greats in the field, François Jacob, who wrote, the probability that a functional protein would appear de novo, brand new, by random association of amino acids is practically zero. It is very small, but that's not to mean to say that it does not happen. And we now have evidence of some of these genes which have occurred, literally bang. You've got a mutation, it's changed a useless piece of DNA into a useful piece of DNA. And these are some, I'm not going to bore you too much with these mechanisms, but they're a number of rather simple models whereby this can occur. So that's kind of amazing if you think about it. So in one generation, this piece of DNA signifies nothing. In the next generation, because of a mutation or mutations, you've got an active gene. You've just created a whole new gene. And this is a horribly detailed slide, and it's mainly about some fish that live in the Arctic. Fish that live in the Arctic have a particular challenge, just as if you lived underwater in the Arctic, okay, I'm not recommending it, is cold, duh. Your danger is that your blood system would freeze up. So just think about on a cold day in Eastern Anatolia, you need antifreeze in your car engine system so that things don't freeze up. Antifreeze is just a way of lowering the freezing point of the fluids in your engine. These fish need antifreeze, and they have a protein-based antifreeze system. And because they've moved relatively recently into this area, we can actually track the invention, if you like, of fish antifreeze in their bloodstream and look at the genetic processes that they've managed to accumulate in order to do this. And that, yes, started from scratch. I'm not sure whether that's enthusiasm or boredom or mere hunger. Yeah, right. That's starting from nothing. Most genes new genes are made from old genes. And this is various processes that cause duplication of bits of DNA. So this is a famous one in meiosis, when you're generating your gametes, eggs or sperm. If you've got, here are mum chromosome, dad chromosome, and you've got two similar genes that sit beside each other, which we'll call L and R. L should pair with L, right? But sometimes if they're too similar, R pairs with L. And then if you get a genetic crossing over event, you're going to create a copy of the chromosome which has an extra copy of the gene and one which has lost a copy of the gene, right? Now, why does that matter? Because of redundancy. Let's think about this. If I've got one gene, let's call it A, and it's performing some important function. If a mutation happens to that gene which changes its function, that's a problem because I need gene A to perform its basic function. Now, if I've duplicated gene A, so I've got a gene A prime copy, and I have a mutation in gene A, I've still got gene A prime that's performing the basic function. So now evolution is permissible. Do you see what I'm saying? because you've got that spare copy. It's the redundancy that permits evolution. Famous example, beautiful example. This is the genes that collectively make up your hemoglobin. This, as you all know, is the pigment in your blood that binds and transports oxygen. It's made up of multiple genes, globin genes. In fact, we understand the history of globin genes. These are your globin genes. You have two families. These are related groups of genes, beta and alpha. And we can actually trace back. This is 300 million years ago, 450 million years, way back when. This is myoglobin. This is an early form of globin, which is used for the storage of oxygen in muscle and so on. These are all subtly different, okay? But they're all ultimately related to a single globin gene that originated early in the history of animals. And the cool thing about this, of course, and you might be familiar with this is a plot of your hemoglobin, your oxygen-carrying molecule in the bloodstream, from pre-birth to post-birth. Now, pre-birth, you had this very awkward experience of being in utero, right? Where are you getting your oxygen from? It's coming from mum's bloodstream across the placenta, right? Then this traumatic event occurs. You're born, bye-bye placenta, bye-bye mother's bloodstream. What you've got is oxygen. You're getting your oxygen now across the lung from the atmosphere. It's a completely different process in terms of oxygen acquisition. So the molecule needs to be thermodynamically distinct before and after birth. So it turns out there are 4 submolecules that go into the hemoglobin molecule. And pre-birth, it's alpha and gamma. Post-birth, it's still alpha, but now you've switched out the gamma with beta. Okay? Subtle differences, and it's all due to that duplication and redundancy that buys you. But that's kind of boring. It's standard. Let's look at a truly dynamic genome, but also this process. And this process is a crazy one. This one also requires duplication and therefore redundancy. When I say it's a crazy one, this is something that happened at incredibly low frequency. But something that happens at incredibly low frequency in evolution can have an impact, because we've got 3.9 billion years to play with, 1. And 2, we have gazillions of individuals. Each one of them could possibly have a mutation, right? And if it's a good mutation, woo, that's picked up by natural selection and increased. But what this is, a documented new gene that arose in fruit flies, my boring little organism, and it's actually an exquisite story. So once upon a time, there was an ancestral gene that duplicated. So now you've got a copy of it. So now we've got the redundancy we need, okay? Now, we're going to go to a completely different part of the genome. This is on a different chromosome. This is a gene called alcohol dehydrogenase. And here's the structure of the gene. Genes, as you maybe know, are split into functional bits, exons, and non-functional bits called introns. I've colored the introns yellow here. And what happens in the production of a protein eventually is we make a messenger RNA from the whole thing, then we knock out the introns. So here we have what we call the edited RNA transcript without the yellow bits. Fine. Now, why am I telling you this? Because once upon a time, back in fruit fly evolution, this thing in a cell encountered a weird enzyme. It's an enzyme used by some viruses, which converts RNA into DNA. It's called reverse transcriptase, okay? So we've got our RNA, and it's met reverse transcriptase, which means it's been turned into DNA. And then, again, another vanishingly rare event, somehow, and I literally mean we don't know, it got parked into the middle of this duplicate gene, okay? So this is DNA, it's all DNA. So what we now have, we still have the functional ancestral gene. We have this new hybrid gene, which is comprised partly of the ancestral gene and partly of the edited transcript of this alcohol dehydrogenase gene. That is weird, okay? What we have, And we've given it, a friend of mine actually discovered this. He named it after a character in Chinese mythology called Jingwei. So the new gene, Jingwei, is cobbled together from the two previous genes. Now, 999 times out of 1,000, you take two random genes and park them together to create a new gene, and you've got a piece of junk. This turned out to be useful, okay? So natural selection liked this and increased it in frequency in the population until it was present in the entire species. Here's Jingwei, by the way. She had a bad experience. She drowned, but she has a sort of resurrection thing and tries to fill the sea with stones and twigs. Sounds depressing, actually, but anyway, each to their own. But that's what I mean about a dynamic genome. Doesn't get more dynamic than that. These weird duplication events, then this even weirder reverse transcription event. And finally, the third piece is parking that piece of DNA into the ancestral duplicate gene. All right. So new genes can come from scratch. They can be courtesy, some duplication process and redundancy. What's horizontal transfer? Horizontal transfer is when you get a gene, not from mum and dad, which is where you get most of your genes, but from a species which might be very different from you, something which is completely different, okay? You've acquired genetic material, not down the family tree, but across the family tree. That's the horizontal piece of what, as I say, we call horizontal transfer of genetic material. And this is a beautiful example. This is a bad guy. This is a white fly. It's not very sexy, I appreciate. It's a pest of crops. We don't like white flies. Now, white flies and, well, all herbivorous insects have long been in a sort of arms race, a fight with the plants that they eat. Because obviously plants don't want to be eaten, but the insects want to eat the plants. So there's what we call an arms race. So the plant produces a toxin that poisons the insect, so the insect can't eat the plant. But then the insect evolves the ability to detoxify the poison, so it's okay, so now it can eat the plant again, and so on and so forth, okay? So what's happened here, it's actually really interesting. These plants have produced some rather toxic chemicals which poison our poor little white flies, okay? Now, so the white flies sort of being defeated in this arms race. What happened? What happened is amazing. So here's the poison. They're called phenolic glucosides. So these things are toxic to the white fly. Now, obviously, the plant has to worry. If it's producing toxic chemicals, it has the wherewithal to not poison itself. So it produces an enzyme, don't worry about the details, called BTMA-T1, which detoxifies it for the plant's own purposes, okay? So the plant is not poisoning itself. So the plant has both the poison and, if you like, the genetic antidote to the poison on board. What's happened? Somehow, and again, we don't know how, ancestrally, some of these white fly have acquired the DNA that encodes the antidote gene, the BTMA-T1 gene, such that they can now detoxify the material themselves. So this isn't a whitefly gene. The whitefly has acquired a plant gene, this baby, in order to detoxify the plant defenses. That is super cool. And by the way, you can manipulate the ability of the whitefly to survive in the face of these toxins using genetic tricks. So this isn't just weird evolutionary biology, isn't this interesting stuff? This actually has serious implications in terms of developing pesticides and so on to confront this menace to our crops. Horizontal genetic transfer. Now you're saying, whiteflies? I don't care about whiteflies. What about us, humans? Well, horizontal transfer in our species, the one weird form of horizontal transfer As I say, it's when instead of receiving my DNA from my immediate ancestors, I get a piece of DNA from a very distant relative. Okay. Now that can be through what we call hybridization between different species. Famous case in point. Everyone in this room who is not a pure African, and looking out, I think there is nobody who's a pure African. I mean, we have someone from South Africa, but I'm not sure I'm counting you, is partly Neanderthal. Why is that? Because when our species evolved in Africa, came out of Africa, and in the Middle East and in Western Eurasia, encountered an established relative species, not especially close relative, the Neanderthals. And we interbred, or our ancestors interbred with the Neanderthals. This was an amazing moment of horizontal genetic transfer, actually, if you think about it, because Neanderthals, think, our ancestors are Africans. They're coming out of Africa, okay? They're adapted to hot weather. They have dark skin. They have encountered various tropical diseases. And now they're moving to central Poland, okay, where it's minus 25 all winter, those kind of conditions. And obviously they're encountering all sorts of different diseases and so on. That's a big step. The Neanderthals also probably evolved in Africa, came out of Africa about 600,000 years ago. So more than 500,000 years ago. They've been in Europe and Western Asia for a long time. They have evolved under natural selection to cope with Polish conditions, to cope with Polish diseases, to cope with the new environment. Now, that's taking generations and generations and generations, right? That's how natural selection works. This individual has, let's say, fairer skin, which is adaptive in a climate like Poland relative to Africa. And that individual leaves a few more children than other individuals. The next generation, a few more children, a few more. I mean, it's just a very slow, inefficient process. It works, but it's slow and inefficient. But then our ancestors come out, Homo sapiens, Neanderthal, a moment of intimacy, as we say. And what does that mean? It means that our species has acquired from Neanderthals all that stuff, all that genetic power to cope with Poland, if you like. We've not had to wait hundreds of thousands of years while natural selection has done its work. We've just got it. Thank you very much. So we see and with the term of art here is adaptive integration, where you're getting horizontal transfer from a distantly related species, but you're getting good stuff. But this is my favorite version of that process. And I've talked about it here before, so I'm sorry if you remember this, but it's so cool. Tibetans are adapted to life at 10,000 feet, at 3,000, 4,000 meters. So if you and I go to where they live in the Himalaya or on the Tibetan plateau, we can barely breathe, right? They have genetic differences from us, which allow them to do that. And in particular, there's a gene called EPS1, which is involved with oxygen metabolism. It's if you like, the key to Tibetan success in Tibet. We know that. The interesting thing about EPS1 is it's very different from every other human, from the non-Tibetan version of the EPS gene. So what's going on? This is a slightly complicated plot, I apologize, but it's actually really cool. This is the gene, EPS1, and the background gray is everything's the same. And so the black blocks are where you've got different mutations. And we've got two populations here, basically. These are local non-altitude adapted Chinese, and these are altitude adapted Tibetans. And all I really want you to get to start with is the top part is rather different from the bottom part. And that's what I mean. There's a whole bunch of differences at this gene between Tibetans and non-Tibetans. The cool thing, you probably can hardly see it. Up here, we have one individual. A Denisovan is a close relative of a Neanderthal. So Neanderthals came out of Africa. They split into pure Neanderthals and the Denisovans. It turns out that the Denisovan gene is the one the Tibetans have. The Tibetans got their adaptation to high altitude from a distant relative, from adaptive introgression. That's so cool. Because remember, it took years and years and years, generations and generations for the poor old Denisovans to undergo this adaptive process. And the Tibetans have said, okay, I'll have sex with you. Thank you very much. Now we've got it. Thank you very much. See ya. This is sort of the process. So here's the gene, and I've colored it showing sort of genetic differences accumulating over time. So this is the Neanderthals leave Africa. So here's the split between the Neanderthals and Denisovans. Sometime in the history of the Denisovans, they adapted to altitude, and then they interbred with one group of modern humans for whom it was advantageous. Now, this is super cool, except there was a problem with this idea, which is the only material we had from Denisovans, this is archaeological material, came from a cave in Siberia, which was at like 200 meters above sea level. So why would you go to the trouble to adapt to high altitude if you live at 200 meters above sea level? I mean, you don't need those adaptations. But then, I think this is so cool, we found more Denisovan material. And here it is. It's not super sexy. It's part of a lower jawbone. But the cool thing about this is, where did this come from? It came from this cave in Tibet at about 11,000, what is that? 3,500 meters above sea level. Right? So, story's exactly right. That's what the Denisovans did. They lived in Tibet, they adapted to Tibet, and therefore met the future Tibetans, and the future Tibetans massively benefited from that EPS1 allele they acquired from the Denisovans. Okay, let's keep going. Accelerated regions. These are super cool, actually. And again, I think I mentioned these in a previous talk here, but it's worth revisiting. How do we identify the key changes that make humans human? I mean, think back. 7 million years ago, we had a common ancestor with chimpanzees. One lineage went off and produced the chimpanzees and the bonobos. The other one produced us. So we're going from a sort of gorilla, chimp-like common ancestor 7 million years ago to this gentleman. What's your name? Dennis. Anyway, that 7 million year journey to produce Dennis. What we'd love to do is identify the particular pieces of DNA, the genes, the key changes that took us from that ancestral state to produce this bipedal. That's a big thing. 2 legs. I mean, walking on 2 legs. Everyone has at least 2 legs. And this very large brain. One way to do this, this is super simple but super cool, is to look for pieces of DNA which are highly conserved. In other words, they're very important in nature, except in our species. Let's have a look. So here's a chunk of DNA in chicken, and here's the same chunk of DNA in chimpanzee. And notice, I think it's 300 base pairs or something. There's 2 differences. Okay, what this tells us is this is functionally important. So mutations which change this get knocked out by natural selection. They're deleterious, they're damaging, they're bad, right? So basically, it's natural selection is keeping everything the same, all right? Except look at that. And this is our closest relative, right? So we've got blah, crazy, parking G's and C's in all over the place. Now, there are various ideas as to what's happened, but these are the big differences that make humans, right? Something which has been conserved, and gets to us, change, massive change. So that implies a change of function. But how can you have a change of function of a critical piece of DNA over such a short evolutionary period? This is really cool. We now know that genomes, yeah, we think of them as linear AGCC, We now know that the 3D configuration in the cell, I know it looks like a bad hair day, these are different chromosomes by color. And so this 3D configuration is actually critical because maybe there are interactions between a green chromosome gene and a orange chromosome gene here. And the fact they're sitting next door to each other is absolutely critical. So with that new three-dimensional appreciation of genetic information, we have this, which I think is beautiful. This shows how one of those so-called accelerated regions could suddenly, almost literally overnight, acquire a whole new function. So what we have here is the chimpanzee situation, and we've got two genes, A and B, okay? And we've also got this, our anomalous so-called accelerated region here. And this is the way, this is our 3D structure, obviously a bit of a squiggle. And at this stage in the human situation, this is associated with gene A, the pale gene here, okay? And then Bluey, one of those transposable elements, one of those dynamic events we talked about earlier, parks itself in there, bang, right? What's that done? It's now changed the 3D configuration. It's changed the length. It's changed everything. And what it's done is it's brought this same piece of DNA into proximity with gene B. So suddenly it's doing something literally night and day different. Not because it aspired to or it wanted to, but because of this random reconfiguring event parking in one of these transposable elements. So I think this is probably going to be the story of where we come from. It's going to be weird because it's going to be accidents resulting from the parking in of random bits of alien DNA like these transposable elements. Okay. Keep it moving. I'm now going to change gears but in a slightly weird way because I've been talking about how dynamic the genome is. Now I want to talk about why the genome is sometimes not so dynamic, and that can be useful from the point of view of telling us important things about the evolutionary process. So just to remind you, genomes are organized into chromosomes. So this is the human chromosome 1, 2, 3, and so on. And as I say, they start large and get smaller until you get to the sex chromosome. So this individual is a male with a large X and a puny Y. And each chromosome, of course, is an enormous, lengthy, even the smaller chromosomes have thousands of different genes on. So this is chromosome 2 again. So this is a long one. Each one of these weird acronyms down here is a different gene, okay? So what we're dealing with is an enormous amount of genetic information strung out along these chromosomes. Okay, so that's the basic thing. We've got a set number of chromosomes. We've got a set of genes on each chromosome. But remember, we have mechanisms, I've already shown you this slide, mechanisms whereby the chromosomes can change configurations through these translocation and so on events. Now, so the question is this: can you pretty much put genes at random anywhere on a chromosome or on any chromosome, as long as you've got gene A? Doesn't matter if it's on chromosome 13 or chromosome 19, right? Or do you have to have gene A on chromosome 13 because it's next door gene B, because in some way that they functionally interact in that way? So let's ask that question in a slightly arcane way. This is a general family tree of animals, and it's a very large-scale family tree. So this is us, for example. This is our group, the chordates, fish and other vertebrates. But you've got all these weird, slightly simple organisms which evolved as early animals, including jellyfish, these so-called comb jellies and sponges and so on. And the common ancestor was on the order of 600 million years ago. And we're dealing with a lot of evolution. So what we're going to do is compare a sponge and a jellyfish genome. Now, this is actually a super cool plot, but it's going to induce a migraine if you stare at it for too long. What we have is on two-dimensional, these are the sponge chromosomes. So chromosome 18, 22, 23, don't worry about the designations, they're arbitrary. And these are the jellyfish chromosomes. And so let's look down here. Every time you've got a colored dot, that is a gene. And every colored dot here implies that the genes on RES7 are present on EMU18. So in other words, all these purple genes in both these extremely distantly related organisms are on the same chromosome. They're in the same genetic neighborhood. So that tells us that probably there's strong natural selection trying to ensure that things don't move around too much, all right? Because there are good reasons for having things in the same place. And notice, you can see events that have occurred in the history of evolution. We're missing material here. It's been moved down here. So it's put on this species, it's all on one chromosome. On the other species, it's on two separate chromosomes. Okay? So you can, another instance of an aberration there. But in general, you've got this really striking pattern up the diagonal. Now, these gray dots are just things which have got randomly relocated all over the place. But what I want you to get is that signal on the diagonal is really strong. So what we're looking at is a situation where apparently there's good reasons for having genes A, B, and C on chromosome 1. You don't want to break up that association. And the term of art is syntony here, where everything is kept in the same order. Now, why am I telling you this? Well, I'm telling you this because it's really interesting. But I'm also telling you this because it's really useful. So we're going to use this to solve a really interesting problem in neuroscience. Or, well, I don't know if it's an important problem, but I think it's an interesting one. We're looking at the distribution of nerves now. Sponges don't have a nervous system. Ctenophores, comb jellies, have this they don't have a head base, they don't have a brain, they just have a sort of network of nerves, as do jellyfish. You, of course, you and spiders and snails and everyone else have a centralized, or most of you have a centralized nervous system with a brain. Now, we have a problem with this phylogeny because we can't figure out which one, the sponges or the comb jellies, are the sort of outside relative of these guys. Now, why does that matter? Because it tells us important information about when, where, and how the nervous system arose. And just in case you're puzzled, what is a comb jelly, sometimes called a ctenophore? They're beautiful. That is one sexy comb jelly, come on. So we've got two possibilities. What is the sister group of all other animals? So here's, if you've got the comb jelly, this is the ctenophore, and here's all the other animals, and vice versa, the alternative. Why does this matter? Because remember, nerve net, sponges, no nerves, okay? Now, so this is simple. We just use our standard tools to reconstruct an evolutionary tree. We can do that by sequencing DNA, lining up the DNA and seeing which is most similar. Now, that's easy to do if we've got very closely related species. So here's a bunch of different lineage, different strains of fungus, and they're all this is the same DNA sequence. There are a few differences, but you know, it's easy to line them up. Now, if you've got distantly related species, there's been so much evolution, there's so much change. These proteins are now different sizes, different shapes, different lengths. It's a mess. So the blacked-in bits are the bits which show some similarity across species. But you've got all these dashes which are gaps, because how else, if you've got one protein which is 430 amino acids long and the other which is 590, you're going to have to incorporate a lot of gaps to do that. And this is basically guesswork. I mean, and I'll give you a nice example, actually. Why is this N? Some computer algorithm has reconstructed this N. These are, by the way, are the single amino acid codes, and put it here rather than here where there's an N here. Don't ask me. So once we're looking at very ancient reconstruction, we've got problems. And you can think about this intuitively. It's easy enough. Humans and chimpanzees are pretty damn similar, and we're pretty similar to cows. But what about these guys? right? So they have antennae. Are those similar, equivalent to ears in cows? I don't think so. And what about, well, but these antennae, are these antennae equivalent? I don't know. You get the idea. Once you've got very ancient nodes in your family tree to reconstruct, you've got problems. Now, the cool thing is that we can use this pattern as a reliable indicator of phylogenetic affinity to do our phylogenetic reconstruction, just as I've suggested. So we're going to use this, and now I'm going to blow your mind. It's sort of almost at the end of time, so this is going to be difficult to digest, but this is super cool. We're going to take this, and this, now we've got 6 species. So this is a comb jelly, jellyfish. This is us. We're bilateria. It's not us, but it's a distant relative. And these are 2 sponges. And each bar is a chromosome. So basically, and this is the material. Each line is a gene on a chromosome. Okay? So basically, on this chromosome, in this sponge, you've got these genes, same genes here. They've moved around a bit here, but they're also on the same chromosome here. Moved around a bit here. Then things get really messy up here, right, for example. And we could do that for every chromosome. I mean, please, it will just get a headache if you look at it too hard, but just admire that scientific illustrations tend to be boring. Graph, some points. Look at that. Now, what I want you to get from this is you can actually identify particular events. So the material which was on this sponge chromosome 9 ended up on this sponge chromosome 9, but then got mixed in with this material from chromosome, whatever it is, 5 down here in the bilaterian here, okay? I'm not going to do chapter and verse on that. I just want you to get a sense so you can actually identify these evolutionary rearrangements, which are rare but important. And therefore, come on, it's beautiful. Therefore, we can solve our phylogenetic problem. So we've got this problem. Let's get rid of that. Let's apply the logic that I've just applied. Ooh. Now this is weird. So now we've figured out the history here, but it turns out that we've got nerve net here, but in between are the nerveless sponges. So what's happened? Well, it seems that we have, it's actually really cool, that nerves have evolved twice convergently in 2 different groups, the comb jellies and in the group that would eventually produce jellyfish and indeed us. Convergent evolution, really powerful evidence of the power of natural selection. And I'm going to finish with, I think, one of the cooler discoveries of the past decade on nervous systems. We all know that animals have nervous systems, or most animals, not sponges, have nervous systems, and it's sort of what makes us cool. We can sit in classrooms and listen, for example. That's courtesy of nervous system. Plants are boring because they don't have nervous systems. They just stand there. I'm a plant. Right, I mean, it's not sexy or interesting, right? But they do. Well, they don't have nervous systems in the same sense. They have convergently evolved what you might call pseudo-nervous systems.
When a bug tries to bite you, you can always swat it away. Unfortunately, most plants don't have that luxury. What they do have, scientists have just discovered, is an innovative system of communicating danger to their other parts, using some of the same signals as in animal nervous systems. Researchers have shown that plants can sense the touch of a caterpillar's tiny toes and respond defensively. Scientists made this discovery as they were investigating how plants sense gravity. They suspected calcium signaling was involved, so they genetically engineered an Arabidopsis to glow brighter whenever calcium levels increased. When they observed the plant under a fluorescence microscope,
it was being damaged.
Response to moving. So how do plants know to go on guard? It starts with a chemical, an amino acid called glutamate. Any type of agitation, from a bug bite to a mechanical cut, causes the release of glutamate, activating receptors that trigger a calcium-based signal that moves through the plant. The signal travels the green highway of the plant's vascular system in just 1 to 2 minutes, activating defense hormones that prepare distant leaves for an impending invasion. By gaining a better grasp of this process, researchers can better monitor signaling and someday may be able to manipulate plant communication. So the next time you brush up against a plant, know that they are feeling your every touch.
Isn't that cool? So what's happening here is you've got a plant and a caterpillar takes a bite out of a leaf out here. That leaf can then send signals all the way through the plant such that every other leaf is prepared for an aggressive invasion by a caterpillar. It's a nervous system, right? So this is convergent evolution. It's obviously very different from the animal nervous system, but it's serving exactly the same function. And that is all I have to say. I hope with me, you feel excited, partly by the technological advances of the science that I'm talking about here, but also by this vision of an evolutionary process that over 3.9 billion years, over all sorts of weird, low probability events, went from that wiggling RNA molecule all the way to Denis here. Thank you very much. And I happen to know do we have time for questions or we don't? We have time for, there's a class coming in here in 3 minutes time. So if there's one question, pressure on the questioner. Yes.
Transfer. I can easily understand Neanderthal human.
Yeah.
Why flat? Is it transferred by eating?
Yes, probably. So remember, this is a very intimate relationship between the plant and the animal. The animal's basically plugged in, sucking the juice out of the plant. And in doing so, of course, it's getting a whole mouthful of plant DNA. All you need is some accident to occur such that some of that DNA ends up in one of the animal's germ cells. Look, these are low probability events, but we do often, the more we look now, when you have especially parasites that live very closely with their hosts, we find there's actually a fair bit of genetic exchange, things just literally leaking from one genome to the next. But that was, if we're going to have one question, that was a great one. Thank you very much for coming, and especially to Tevitol and Koch. Great to see you guys.